So we're supposed to be on chapter 3, I guess, cells, and we're going to need your help to remember where we left off. Um, Teaching a bunch of different classes this semester that all doing similar things, and I can't keep track of. I should remember to write down at the end of class where I stopped, which slide number, but I never remember to do it. But, so we're supposed to be on chapter 3, so we did this already. And, but it says cell structure and function lab. I have a couple little lab things to do over there. Not a big deal. Won't take a whole lot of time. We can play with that a little bit. And I'd like to start playing with that anatomage thing in this class. Use that for spend some of our lab time doing that. I'm still learning it. I feel like I know about maybe 5% of the functions of that thing. It's the more I look at it, the more amazed of how much stuff is in that thing. It's there are quizzes and exams built into that. I don't know how to use them, but and this is my last semester because I'm retiring, so I'm probably not going to spend as much time learning that thing as if I were staying, because it's not worth investing a whole lot of time learning something I'm not going to be using very much. Okay, anything else we need to talk about? Oh yeah, there's going to be, starting next week, something we did last semester. We're going to do it again. It's called Bio Lab. Like, they have a math lab and an English lab here, and this is going to be called the Bio Lab. And it's just time every week in room 133, the general biology lab across the hall and down. There at certain times during the week there will be a biology instructor in there for any biology student taking any biology course at this college can go in there and get help. So I'm going to be in there 3 to 5 Monday afternoon, 3 to 5. So if you need help tutoring or anything, or having trouble with anything, come on in. I'll just be sitting there all alone. Last semester, I never had one customer all semester. And it was last semester we did just one. I had to do just 1 hour a week. Now they're say we need to each do 2 hours. So I'm going to be sitting in there for 2 hours a week all alone. I usually work on something else, but I know there's biology students who need help or could benefit from help, and I wish they would come. So if it's not me, there'll be somebody else, and there won't be somebody in there all the time, because there's only 5 of us, I think, biology instructors. So, but I think there's going to be somebody in there every day, at least part of the day. Okay, so chapter 3. Where are you? Here. So did we started this, right? Anybody remember? Or did we start this chapter? So what did you say?
I know we spoke about flagella.
Flagella, okay. Well, that helps.
So that's when slide 37 we should start, unless y'all think.
Okay, so we were talking about parts of the cell, right? And talking about this, and so we got this far, and we talk about all this stuff.
I don't remember that part, but
No,
I
that was after you fell asleep, right?
Yeah, it was kind of blurry.
Okay. Well, we'll just pick it up around here somewhere. So we're talking about parts of cells, and um, yeah, you guys don't have to know too much details about this stuff, just basically. But the flagella, that's the tails. The tails. Some cells have tails called a flagellum, and the only human cell that has that is a sperm cell, and it uses that for swimming. Right? It moves like a corkscrew to push it through the liquid environment um, on its way toward where there might be an egg waiting for it to fertilize. Okay, centrioles. This is part of that cytoskeleton. The cytoskeleton is just a bunch of protein fibers running through the cell, holding organelles in place, um, and some of those cytoskeleton filaments can get longer or shorter to move stuff around in the cell. And the centrioles are they only function when a cell is dividing. Um, so uh, when a cell divides, it splits in half and into two cells, and the cell that divides has chromosomes in it, and so chromosomes have to be moved into the two daughter cells, and that's what the centrioles do. They move the chromosomes during cell division. The rest of the time in a cell's life, they don't do anything. They're not functioning. Ribosomes, you just need to know that this is where the proteins are made. And we'll see a little later in this chapter a little bit more about how the ribosome makes protein molecules using information that's in genes. Um, okay, the endoplasmic reticulum, the ER. This is just need to know this is where new molecules are made in the cell. And there's two kinds, rough ER, smooth ER. Let's look at a picture. So rough ER shown here, it has all these little dots on it. That makes it look like sandpaper. That's why it's called rough ER. Those little dots are ribosomes for making proteins. And so the rough ER is where proteins are made because it has ribosomes on it. Not all the ribosomes are on the rough ER. There's a bunch of other ribosomes just out here floating around in the cytoplasm. The smooth ER does not have ribosomes for making proteins, so this makes other kinds of molecules, carbohydrates and lipids, fats. And one of the kinds of lipids that's made by the smooth ER is phospholipids, because that's what the membranes of a cell are made out of. So if the cell needs more membranes, then the smooth ER would make more phospholipids for that. So this is where it says nucleus. This is nucleus. So the ER is very close to the nucleus, because the nucleus is where the information is for how to make those molecules. The information is stored in the genes, which are in the nucleus. Okay, the Golgi apparatus. Some books call it the Golgi complex or just the Golgi. I think Golgi was a guy's name who discovered it or something. So so the ER makes these new molecules. The Golgi finishes making those molecules. It finishes putting them together, and then packages them to move them so they can go to wherever they're needed in the cell or outside of the cell. Some cells make stuff to be released out of the cell, like the cells of glands. We have glands that make hormones, glands that make sweat, earwax, tears, saliva. the cells in those glands will make that stuff with the ER and the Golgi, and then it'll be released out of the cell. So here we see the ER again, the rough ER and the Golgi. So the ER, it would make a protein molecule, put it into this, called a vesicle. It's this little sac made of membrane. And then that'll take it to the Golgi. The Golgi will finish making those molecules. And then the Golgi puts it into another vesicle. And now it's ready to go wherever it's needed in the cell, or in this case, you can see it's releasing to the outside, out of the cell. And this stuff, this is all made out of membrane material. The same kind of membranes as the plasma membrane that surrounds the cell makes up these, they're like hollow, flat sacs or tubes made of membrane. Okay, mitochondria. This one's important. They're called the power plants of the cell. But, a power plant is where a fuel is, like coal, is burned to generate electricity or something. And in the mitochondria is where those chemical reactions happen that break down nutrient molecules like sugars to get the energy out of them, the energy that the cells use. Those chemical reactions are called cellular respiration, aerobic cellular respiration. Aerobic means it requires oxygen. So that happens in those mitochondria, and they're called the power plants of the cell because that's kind of like burning sugar. Because burning is just a chemical reaction between a fuel, like coal or gasoline or something, and oxygen. And that chemical reaction releases heat energy. This is kind of the same things, like sugar, chemical reactions to break down sugar using oxygen. But it's not to get heat energy. The energy is stored in these ATP molecules, like these little rechargeable battery cells, rechargeable batteries in cells. And then that'll be the energy that the cells use for most of the things that cells need energy for. And tells us here the cells of our body that use a lot of energy, like muscles. It takes a lot of energy to do this, for a muscle to make your body move. And so those cells of our muscles have a lot of mitochondria. Always have to be making a lot of ATP for energy for moving our bodies. Brain cells have pretty much mitochondria. Takes pretty much energy to think, I guess. So that's what a mitochondria look like. They're long ovals, or like a sausage. Okay, a couple other organelles, lysosomes and peroxisomes. These are both do similar things, called the garbage disposal of the cell, because their function is to break down molecules that the cell doesn't need, or organelles, old organelles that the cell doesn't need. The lysosomes will break them apart, break them down. Even some molecules that might be harmful or toxic will be broken down by these. This lyse means to break something down. You'll see that lyse in other words, like this word. What does hemo mean? Blood. Hemolysis means if red blood cells are breaking apart, it's called hemolysis. And peroxisomes, that name comes from because it produces hydrogen peroxide as a byproduct from breaking down other molecules. So they're called peroxisomes. What else is important? No, the nucleus, of course. So this is where the chromosomes are. Here's a picture of a nucleus. You cut open to see the inside, but it's so under a microscope, you look at cells under a microscope, you can usually see a dark spot that will be the nucleus. And inside the nucleus, there's a darker spot called the nucleolus. This is where the ribosomes are made. Remember, the ribosomes are where the protein molecules are made. So the nucleolus makes the ribosomes, and then the ribosomes leave the nucleus. So there's ribosomes, these little dots are ribosomes. The nucleus is surrounded by a double membrane, so 2 layers of membrane, and that's called the nuclear envelope. And it has holes through it called nuclear pores. So that's, for example, that's how the new ribosomes that are made by the nucleolus would get out of the nucleus through those holes. This is all chromosomes here. Chromosomes, we did the DNA thing with strawberries last week, right? Mm-hmm. So those are very long molecules, but they're all coiled up inside the nucleus like that. Okay, chromosomes. A human cell has 23 pairs of chromosomes. Other species have different numbers, like a dog, I forget what it is, but a dog might have 19 pairs of chromosomes. A horse might have 27 or something, but for humans, it's 23 pairs. And when somebody in a lab arranged them this way, they're not normally arranged this way in a cell. And an image, when somebody arranges the chromosomes like this and takes a picture of it like that, that's called that word's not on here, I guess, but that's called a karyotype. Karyotype. Just a picture of all the chromosomes laid out like this. And the chromosomes are numbered, and from the number 1 is the longest chromosomes, and then they get shorter and shorter. And the 23rd pair is called the sex chromosomes, and that's the X and Y chromosomes. Anybody know if this karyotype would be from a male or female? Female? Females don't have a Y chromosome. Only males have the Y chromosome. What would females have? 2 X's. Yeah. So this is a male. All of the others are called autosomes or autosomal chromosomes. They're just those chromosomes that are not the sex chromosomes, X and Y. So on any pair of chromosomes, they have the same kinds of genes on both of them. So if there's a gene for eye color right here on this number 1 chromosome, there would be another one in exactly the same place on the other number 1 chromosome, another eye color gene. But they might not be the same. This one might be for blue eyes, and this would be for brown eyes, or they could both be for blue or both be for brown. And that location is called the locus, the gene locus on the chromosome. If this was a female, is there a picture of a female? No. If this was a female with 2 X chromosomes, they would both be the same size, and they would both have the same kind of genes on them But because this is a male, obviously the Y chromosome is much smaller than the X chromosome, so they don't have the same kinds of genes on them. Um, they do have the information, the genes that determine male or female. And the X chromosome is bigger because it has, whether you're a male or female, like I have X and Y, but um, when I was a little embryo and my reproductive organs started to develop, the information for that was from the X chromosome. So in an early embryo, you can't tell the difference between a male and female. Even though the reproductive organs are starting to develop, they look the same in males and females. But at some point, if there's a Y chromosome, this information in the Y chromosome will direct the development of those reproductive organs to become male instead of female. So instead of becoming ovaries and uterus, it would become testes and prostate gland and the male organs. What else? Okay, so it's one from mom and one from dad. So each of us has one chromosome that we got from our father and one from the mother. So um, if you are male, then you got your X chromosome from your mom and your Y chromosome from your dad, because only your dad had a Y chromosome. Your mom didn't have one. But if you're female, you got one X from your mother and one X from your father. And there are some kind of genes on these X and Y chromosomes. For example, color blindness. I'm red-green colorblind, and I know I got that from my mom. I didn't get it from my dad because the genes for color vision are on the X chromosome. And I know I got my X from my mom because I got my Y from my dad. So it's impossible for a male to inherit color blindness from his father, because the gene for that is on the female. He can only get it from his mother. So even if my dad was colorblind, which he wasn't, but even if he was, I didn't get it from him. So I couldn't say, "Damn you, Dad! I hate being colorblind." It was from my mom. Yeah.
So what does blood type come from?
I don't know which chromosome it's on, but it is genetic. It's so I don't know.
Is it from your mom or your dad?
Oh, both. They're like they're it's on one of these chromosomes, because there are two genes that determine your blood type. And I wish we had time. Maybe we will at the end of the semester. We could do some genetics, but there are. Oh, if like for example, there are four blood types, right? There's A, B, AB, and what's the other one? O, right? Um, in order, for example, in order to be type AB, you have to get an A gene from one parent and a B gene from the other. Uh, and both parents determine your blood type. So if you get like two A genes, an A gene from your mother and an A gene from your father, your blood type would be A. If you get two B genes, your blood type would be B, but You have to get one from each parent, an A from one parent and B from the other to get A type AB. If you don't get either gene, you don't get a gene for A or B from either parent, then you're type O. It's kind of complicated, but just like your eye color depends on what you get from both parents. Is it like there's we call and this is different from blood type. I don't want to get confused, but in eye color we say we use the big B gene is for brown, and little b gene is for blue eyes. So if you get a big B from your dad and a big B from your mom, then you'll have brown eyes. If you get a big B from your dad and a big B from your mom, you'll have, little b. So if this is the genes you have, two big B's, then you have brown eyes. If you get two little B's, one from each parent, then you're blue. But what if you get this? It's still brown, because we use these capital and small letters in genetics, because capital means dominant, and this means recessive. That means if you have one of each, the dominant one is the one that determines the trait you have. So this would be brown eyes also. So, I don't know, genetics is fun. Maybe we'll when we do reproduction and development at the end of the semester, we can spend some time doing that stuff. Okay, DNA. How does DNA work? We know it's for storing information, and that's what genes are. A gene is just a section of a DNA molecule. So here's a chromosome, big long DNA molecule. These are, remember, nucleic acid molecules are made up of smaller molecules called nucleotides, and we call them by the letters A, T, C, and G. So that's what this is. Just a long list of letters: T, A, T, 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 C, G, G, A, T, T, and so on. And these can be like hundreds of thousands of these letters long, and so just as one section, there's many genes along a chromosome. So a gene is just a section. Each section is a bit of information, and usually that information is how to make a protein molecule. When a cell needs to do something, like have some chemical reaction happen, it'll make a protein molecule that'll cause that chemical reaction to happen, using information in a gene. So this is called the genetic code, just a language of these 4 letters. DNA are always, remember, DNA is 2 strands of nucleotides side by side. T and A are always together side by side, and G and C are always together side by side. So we say those are complementary. So there's a diagram of DNA again. It's shaped like a twisted ladder. So this would be a nucleotide here, nucleotide here, nucleotide. So C and G always next to each other. A and T always next to each other. Should never be a T next to a C or anything else. If that does happen, it's a mistake, and mistakes do happen in DNA. That's what a genetic mutation is, just a mistake in the DNA. Okay, the cell cycle. It's just stuff that happens during the life of a cell. We call it the cell cycle. And we can divide it into two parts, and it's shown in this like a pie chart here. The different stages in the life of a cell. So this is all called interphase here. All of this is interphase. That just means when the cell is not dividing. And this M phase is just when the cell is dividing. So it's kind of like your life. You have childhood, and then you have adulthood, and you have old age, and you also though have a period of time when you might reproduce, make babies. So this is when the cell would be making baby cells, like dividing. The rest of the time, it's doing other stuff. So G, this G phase stands for growth. G is for growth. So after a cell is formed, so a new cell forms by cell division. So we'd say this is the beginning of this cell's life. When its parent cell divided, that's the beginning of its life, which would be right here, right after the cell division. So this brand new cell, it's only about half the size of a normal cell of that kind. So it has to grow to get to be the normal size. So that's what happens in the G1 phase, just growth. And then S stands for synthesis. Synthesis means to make something. And in chemistry, it means to make molecules. The molecules that are made in the S phase is DNA. So this would be when this cell is getting ready to divide. So it's all grown up. Now it decides I'm going to reproduce. I'm going to divide in half. So in a human cell, there's 23 pairs of chromosomes. So if this cell has 23 pairs of chromosomes and it's going to divide, these cells have to also have 23 pairs of chromosomes, right? So all the chromosomes, every chromosome in this cell before it divides, all these chromosomes, they're all going to be copied. A copy will be made of every one of those chromosomes. That's what happens in the S phase. That way, when the cell divides, these will separate and go into the two daughter cells, and each daughter cell will have the full 23 pairs of chromosomes. So now after the S phase, there's all those 23 pairs of chromosomes have been copied. The original and the copy are still stuck together. They stay stuck together for a while. But then this G2 phase, another growth phase, because this cell is getting ready to divide, so it'll grow a little and maybe make some more organelles, maybe make some more mitochondria and stuff, so that when it divides, these daughter cells will have enough organelles, and they'll be big enough to get started in their lives. And then it goes ahead and divides in the M phase. The M stands for mitotic or mitosis cell division, and we'll talk about what that means in a minute. So this says G0, this G0 phase. That just means when G0 is like after this G1 phase, the cell's done growing, but it's not divided yet. So it'll just go into this G0 phase. So it's just when a cell is just living its life, it's not ready to divide. It might never divide. There's some cells in our body divide all the time, like your skin cells. We're always making new skin cells. So those cells divide a lot, but muscle cells, they don't divide. By the time you're born, you have all the muscle cells you're going to have. They never will divide. Nerve cells, some kinds of nerve cells, like of your central nervous system, your brain, and your spinal cord. If you have a traumatic brain injury or a stroke and part of your brain dies, are your other brain cells going to divide to grow back that part of your brain? No, those brain cells, they don't do this. So they just go into G0 phase and they stay there for the rest of your life. They just don't divide. But some cells, they'll be, something will cause them to divide at some point, like maybe an injury will happen, and you'll have to regrow that tissue, and so those cells will leave, come out of their G0 phase and then go into an S phase and G2 phase because they're going to divide. Okay, so interphase, we talked about all this stuff. Okay, a little more about DNA and chromosomes. So these are very, like I said, these are very long molecules. It can be an inch or two long. And so this is our DNA molecule. Here we see the nucleotides or the letters and the complementary A here, T here, T here, A here, G, C, A, T. So these two strands of nucleotides of this DNA molecule, they have the same information in them, because if we know this, then we also know this, right? If I tell you one strand is ATGAC, you can tell me what the other strand is, right? Because they're complementary. So, so this is what's stored in your cells, these long DNA molecules. But when a cell is going to divide, and these chromosomes have to move into the two daughter cells, that would be really hard to do if these DNA molecules were all strung out in there, because these would be like this long. You got 23 pairs of these chromosomes in here, and you're trying to untangle them and pull them into the two daughter cells. It would be really hard. So when a cell's getting ready to divide, all the DNA of all the chromosomes, it coils up like this, like a spring, and then that spring coils up again, and so it ends up being, instead of being these long, very long threads, it looks short and thick like this. And you can see them under the microscope. So if you see chromosomes under a microscope that look like this, short and thick, and they're it's like a letter X, because remember, these have all been copied. These chromosomes have all been copied, and the two are still stuck together because it's getting ready to divide. And these histones are protein molecules that are attached to the DNA along this molecule, and their function is to make it coil up like that, to make it short and thick, so it's easier to move the chromosomes into the daughter cells when the cell divides. Okay, so I said that M phase, M stands for mitosis or mitotic cell division. So there's two kinds of cell division in human cells, mitotic and meiotic, or mitosis and meiosis. Oh, sorry.
One of those units was on the test, so is that extra credit or?
Huh?
Yeah, the DNA.
And we didn't cover that, or what?
There was stuff about how to parent the in the PowerPoint.
There was stuff about.
In one of the chapter, one or two PowerPoints, there were stuff about how to parent the, like, A goes with T.
Yeah, was there a sample question about that too? Or- I usually don't put questions on the exam that aren't in the sample questions, but, so that's what you're saying, there was something on there that
Yeah.
we didn't cover yet? I'll look at that. Yeah, if there was, if I put something on an exam that we didn't cover yet, that, I didn't mean to do that. And if that's the case, I would give everybody credit for that. So I'll check it. So, thanks for telling me about that. I'll look at it. But, so two kinds of cell division that happen in humans. Most of the cell division is the mitotic kind. And that's for just cells dividing to make more cells for growth. You know, when we're growing, we're always making a lot more cells by this mitotic cell division. When you heal, some tissue cells get killed off or destroyed, and like you cut yourself, so some of that skin is damaged. Uh, there'll be mitotic cell division to replace those skin cells as in the healing process. And also, there are some kind of cells, like skin cells, that are always dividing anyway, and just to replace ones that rub off or fall off. Your red blood cells, we have billions or trillions of them, but each one only lives about 4 months, and then they wear out and die. So we're always making new ones by this mitotic cell division. Meiotic cell division is reproductive. That's only for making sperm and egg. So it only happens in the testes of males and the ovaries of females. And the difference between those two kinds of cell division is in the mitotic cell division, the if this parent cell had 23 pairs of chromosomes, then the daughter cells would each have 23 pairs of chromosomes. They would be the same. If this was a skin cell, this would be a skin cell, just like the parent cell, and so on. But in meiotic cell division, the parent cell would have 23 pairs of chromosomes, that like a cell in the ovaries that divides, but then the daughter cells are eggs, and those daughter cells have only one set of 23 chromosomes, not 23 pairs. So this would be 23 pairs. But these daughter cells would just be 23 chromosomes. And that's because if an egg has 23 chromosomes, just one set of chromosomes, and the egg has one set of 23 chromosomes, then when the sperm fertilizes the egg, now that fertilized egg has the normal 23 pairs of chromosomes. So the only human cells that have just one set of chromosomes are the sperm and egg, and they're made by this meiotic cell division. These words, mitosis and meiosis, we use those words to mean cell division, but what they really mean is division of the nucleus of the cell. And the division of the rest of the cell is called cytokinesis. So mitosis, I don't want to go into too much. I don't want to require you to learn too much details about cells, because it's I don't think for most of you, that you're not going to need that for your professions. But the mitotic cell division and meiotic cell division is similar, but mitotic cell division, the process of a cell dividing, can be divided up into these phases: prophase, metaphase, anaphase, and telophase. And so this would be division of the nucleus, these 4 stages. And then the rest of the cell, division of the rest of the cell, we said it's called cytokinesis. So what's this look like? Um, okay. This is. They said there was four. Sorry, they said there were four stages: prophase, metaphase, anaphase, and telophase. But this diagram, can't remember if this is one from the current textbook we're using, but it shows 5. It shows an extra one in between prophase and metaphase. It's called pro-metaphase, but most books combine these together and just call it prophase. I don't know why some books have a 5th one like that. It's just confusing to do that. But anyway, what happens? So here's a cell that's getting ready to divide. So the chromosomes condense and become visible. That means the chromosomes, which are long threads, they coil up to become shorter. And you can see them under a microscope like this. They look like the letter X. Then it says spindle fibers emerge from centrosomes. So these are the parts of the cytoskeleton that I said function only to move chromosomes around. So that's what these things are. These are called spindle fibers, but they're just protein fibers that are reaching out, and they will connect to the chromosomes and move them around. So the nuclear envelope breaks down. So the membrane around the chromosomes will disappear temporarily. And so these centrosomes move toward opposite poles. So these things that are sending out these protein fibers are moving away from each other. So they'll be at opposite ends of the cell, like they are in the next step. So now these spindle fibers they're coming out, attaching to the chromosomes. And then these spindle fibers will move the chromosomes so that they're all lined up right in the middle, all in a row, all chromosomes. That's called the metaphase plate. This this imaginary center line of the cell where all the chromosomes line up. So this stage is called metaphase. Just remember, middle. The chromosomes are in the middle during metaphase. And remember, these are duplicated chromosomes here. So there's like 2 identical chromosomes stuck together here. At this point, they're called sister chromatids. These 2 chromosomes that are stuck together. So now the spindle fibers pull them apart. So now they're separating and moving toward opposite ends of this cell that's dividing. So that's called anaphase. So if you see some daylight in between these 2 lines of cells, or 2 lines of chromosomes, that means that cell is dividing and it's in anaphase. Then when these 2 sets of chromosomes get all at opposite ends of the cell, now a new nuclear envelope will form around each of these sets of chromosomes. So a new nucleus. Each one of these will have 23 pairs of chromosomes, just like the parent cell. And so that's called telophase. And when telophase is happening here to the chromosomes and nuclei, cytokinesis is happening to the rest of the cell. So it pinches in half like this. So that's the rest of the cell dividing in half. Now there will be 2 new cells. Each one has a nucleus with the same 23 pairs of chromosomes as the parent cell has. So these are diagrams. These are micrographs, pictures under a microscope. So here you see all the chromosomes lined up in the center. Now you see some two groups of chromosomes separating from each other, and now they're fully separated in telophase.
We have to know this.
Oh, you don't need to know everything that happens here, but what could be on exam is, what's the, um, what are the phases of mitosis in correct order? And so just remember, um, in biology class, remember IPMAT. Interphase, which is when the cell's not dividing, but then when the cell is dividing, there's prophase, metaphase, anaphase, telophase. Just IPMAT. Okay, there would be micrographs, images under a microscope. So we'll probably, maybe next week, maybe we'll get out the microscopes and look at a bunch of cells, and we'll see that some of them, um, many of the cells will be able to see a nucleus. If you can see a nucleus with a nuclear envelope around it, then that cell is probably not dividing. But because in prophase, as soon as it starts getting ready to divide, that nuclear envelope disappears, and you can see the chromosomes. Then they all line up in the middle, metaphase, remember meta, middle, and then they start to separate in anaphase, and then they're fully separated, and you can see the cell is starting to pinch in half here. So it's almost finished. But then a new nuclear envelope will form around each of these sets of chromosomes, new nucleus. So you can look at tissues of like human tissues and see. You can look around at a bunch of cells and see, oh, there's one in interphase, there's one in metaphase, there's one in anaphase. We might do that sometime. Okay, so meiotic cell division to make sperm or egg is similar, except the chromosomes are copied when it's getting ready to divide, but then it will divide, but there are two divisions. So the cell, this looks like just the same as it would be in mitotic cell division. All the chromosomes are copied, but then the cell divides. But instead of these two copies separating from each other into the daughter cells, the two number one chromosomes separate. One number one chromosome goes into this one, the other number one chromosome goes into this one. Same with the number two chromosomes. This shows only 4 chromosomes. There would actually be 46 if this was a human. But so now each of these daughter cells divides again, and then in the second division is when the two copies separate from each other. But because there's these two divisions, ends up being just one set of chromosomes in each daughter cell. And so if this was happening in the testis of a male, each of these daughter cells would grow a tail and become a sperm cell. The same kind of thing happens in females, except in females, again, start out with a cell in the ovaries, copies its chromosomes, and there's one division and 2 daughter cells, but this daughter cell dies. It's called a polar body. And so, but then this one survives, and it divides again, second time, and then this daughter cell dies, and only one remaining daughter cell that becomes the egg. But it's again got just one set of chromosomes, just like the sperm.
So if it was twins, would both of them survive? If they were twins?
No, that's not how twins happen. Twins, there's identical twins, and then there's non-identical, or called fraternal twins. If it's fraternal twins, this just two eggs got fertilized by two sperm, just like any two brother and sister. They just happened at the same time. If accidentally at ovulation two eggs were released instead of one, and they both got fertilized, that would cause twins that are not identical. But identical twins happens when this egg gets fertilized, and then after that fertilized egg starts dividing and growing into a little embryo, and it's just if it's just a cluster of like 16 eggs, and it breaks in half, each half can grow into a new complete person, because none of those cells have yet started to become any particular type of cells. They're not skin cells or muscle cells or nerve cells or anything yet. They're just cells. And so if you break that cluster in half into two clusters of 8 cells, each of them can grow into but they'll be genetically identical, so they'll be identical twins. So this would be called the first polar body, and then the second polar body. We'll talk about this again when we do the reproductive system. Okay, this word differentiation just means a cell becoming a particular type of cell. Like what I was saying, in an early embryo, those cells aren't any special kind of cells yet, but after as an embryo starts to grow and it starts to look like a person, it's because some cells are becoming muscle cells, some are becoming bone cells, nerve cells, skin cells. That's called differentiation. And that's all controlled by genes. Okay, stem cells. A stem cell is just a cell whose only job is to divide to make more cells. And we have stem cells in our bone marrow for making red blood cells and white blood cells. Those stem cells, all they do is they just stay in your bone marrow all the time, and they divide to make more red blood cells and white blood cells. And well, these terms pluripotent and multipotent, that just means a stem cell that when it divides, the daughter cells can become different kinds of cells. So these stem cells in our red marrow are multipotent because when they divide, their daughter cells can become any of these different kinds of white blood cells, or they can become red blood cells, erythrocytes, all from one kind of stem cell. We have cells in our skin that just divide to make more skin cells, but they're not multipotent or pluripotent, because when they divide, all they are making is skin cells. But these can make lots of different kinds of cells. When someone gets a bone marrow transplant, what they're getting is stem cells replaced. Okay, so there are a lot of different kinds of cells in a human body, and they're different shapes and sizes and functions. There's just a few of them there. And as we go through this course and looking at the different kinds of tissues and organs, we'll see a little bit more about that. A gene, again, a section of a chromosome with information for, usually for making a particular kind of protein. When the information in a gene is used to make a protein, that's called gene expression. So it's like expressing the information in the gene by making a protein. And let's see. Well, talk a little bit more about this and then look at that lab stuff. Um, uh, so again, the genetic code is these 4 letters in a row, A, T, C, and G in some order. That's how the information is stored. And remember, a protein molecule is a long chain of smaller molecules called amino acids, right? And there's 20 different amino acids that make up the proteins. So every protein has those amino acids in a particular order. And that order of the amino acids in the protein molecule is determined by the order of the letters in the DNA of the gene for that protein molecule. And the proteins molecules are made at ribosomes. So ribosomes take the information in the gene and they put the amino acids together in the right order. But what the genetic code is divides up these letters into three letters. So the ribosomes look at 3 letters in a row. You don't look at 1 letter at a time. They look at 3 letters, and then another, and then the next 3 letters, and then the next 3 letters. And in DNA, 3 letters in a row is called a triplet. And each triplet is the code for one of the 20 amino acids. But remember the chromosomes stay in the nucleus all the time, but the ribosomes are out in the cytoplasm. How does the information get from the chromosomes to the ribosomes? It's summarized here. There's 2 steps in this making protein molecules: transcription and translation. So the information goes from the DNA, and then that's copied as RNA, and then that information is used to put the amino acids together to make the protein molecule. So this copying DNA as RNA is called transcription, and then using the RNA information to make protein is called translation. This word transcription, do some of you guys taking the medical terminology course? You taking it? So you know what a medical transcription is, right? That's somebody whose job is to take the medical chart of a patient, or the doctor with the doctor's notes, or some doctors talk into they record after they meet with a patient, they'll talk into their phone or something about what they diagnosed and prescribed for that patient or something, and then at the end of the week or something, the transcriptionist will take that and type it up into a spreadsheet or a Word document or something. It's called transcription, just copying the information from one form into another. So that's what this is, copying DNA as RNA. Translation is converting information from one language to another, like English to Spanish or something. And so it's converting the language of these nucleotide letters, A, T, or A, U, G, and C, into the language of amino acids, the sequence of amino acids. So DNA is copied as RNA. The RNA leaves the nucleus, goes out to a ribosome, and the ribosome makes the protein. So transcription happens in the nucleus. Translation happens at the ribosome. So what this would look like, an example, this would be a DNA, the gene. The 2 strands of nucleotides or letters, and they're complementary, A and T, T and A, G and C, and so on. So the cell wants to make a protein using this gene, information in this gene. but this has to stay in the nucleus, so it'll take one strand or the other. Doesn't matter which one, because they both have the same information, but they'll copy this upper strand, make a molecule of DNA as a copy of this, and the nucleotides in the RNA will be complementary to the nucleotides in the DNA. So there's a T. So in RNA there'll be an A. There's an A in the DNA. Remember, there's no T. There's U instead of T in RNA. So that would be a U. G or C, G, G, C, and so on. So that's transcription. Copying this as complementary nucleotides of RNA. And this is called messenger RNA, this kind of RNA, or mRNA for messenger. And now this will leave the nucleus and go out to a ribosome for the translation. So, and here's where the ribosome looks at 3 in a row. The first 3, AUG, that stands for the methionine amino acid. CCG means proline, GAA, this one, and so on. So these are just abbreviations, the first 3 letters of amino acids. So this would be the protein molecule. These amino acids in that order, determined by what was in that gene. Okay, so we said three letters in a row in DNA is called a triplet. Those 3 letters in a row in RNA is called a codon. So the triplets are transcribed into codons, and then each codon is code for a particular amino acid. So here we see making an RNA transcript or copy from, so the 2 strands of nucleotides of DNA will, like, they separate, or they unzip from each other temporarily, and then it's transcribed, and then they'll zip back together, and then the RNA molecule will leave the nucleus. So that's called messenger RNA. There's 2 other kinds of RNA involved in this translation. Ribosomal RNA, that's just what ribosomes are made out of. It's called ribosomal or rRNA. tRNA or transfer RNA, that's what delivers the amino acids to the ribosomes. So there's all these different kinds of transfer RNA. Each one is for each type of transfer RNA is for one codon, and transfer RNA molecules like this one. This is a transfer RNA molecule. It goes out into the cytoplasm and finds the amino acid, and then delivers it to the ribosome. And so that's what's happening here. This transfer RNA delivering this amino acid to the ribosome. This blob is the ribosome. This is the messenger RNA with the codons. And so for this codon, UUU is for this amino acid, and this transfer RNA, this is called an anticodon. There's 3 letters that are complementary to that codon. So this is how the right amino acid is delivered for that codon, and then it'll be added on to this amino acid. Now there's 2 of them. Now a 3rd one's coming to be added on. So transfer RNA keeps delivering amino acids that will be added to this growing chain of amino acids that will become the protein molecule. Simple, right? So this is all the codons and the amino acids. So so you can see each amino acid has more than one codon. This one has 4 codons they all stand for the same amino acid. Same here. This one has only 2 codons that stand for the same one. And some of them, like this one it says it stands for this amino acid, but it's also the start codon. So that's the start codon, AUG, is what tells the ribosome, here's where you start putting amino acids together. So this, there you see AUG, that's the start codon on this messenger RNA. So when the ribosome is like moving along here, reading codon, and when it first sees AUG, it says, okay, here's where we start adding amino acids. So the first amino acid will always be methionine because that's codon is the start codon. And a couple of these codons, or 3 of them, are called stop codons. So when the ribosome is reading along the messenger RNA and it gets to one of these codons, that tells it stop. This is the last amino acid. Don't add any more. This protein molecule is finished. So here's a summary of what happens. Chromosome in the nucleus, our messenger RNA copied or transcribed from that. Then that leaves the nucleus, goes out to a ribosome. So here's the ribosome reading from one end of that messenger RNA to the other, just reading codon. The transfer RNA are delivering the amino acids, which will be added one at a time to make this long chain. That would be the protein molecule. So this is going on in our cells all the time. Our cells are always making proteins. Many of them are enzymes, which cause chemical reactions to happen. And chemical reaction might only need to happen for a few minutes, and then that protein's not needed anymore. It'll just be broken apart. The amino acids will be recycled and make more proteins. Just a summary of the organelles and cell parts. Okay. A lot in that chapter. So, we didn't even, okay, I'm confused. As usual, I'm confused. So we didn't, I thought in this chapter we did a lot more about oh yeah, we talked about we talked about diffusion, right? And osmosis, okay. Because this little lab experiment we got over here, let's just everybody come over here. So, really, Dan, I maybe should have started this an hour ago and then come back to it. Because at least it takes time for this to happen. Maybe we'll just get it ready today and we'll just leave it sit for a week and see what's at a week from now. But this is an osmosis experiment. Remember, osmosis is water diffusing through a membrane because of difference in concentration between the two solutions on either side of that membrane. One way of demonstrating this is a common lab experiment of osmosis is use this stuff called dialysis tubing. Was it you, dialysis guy? That is dialysis tubing. It's in plastic tubing. I got some soaking in water here, but it comes flat on a roll here, and you have to put it in water to soften it up and let it, so you can open it up like a tube. And that's been soaking for a while. But anyway, like these pieces are about 5 to 6 inches long, and so we're gonna make like little balloons out of them. They're, um, balloons like look like sausages, and so we need to tie off both ends of that tube. And we got these little rubber bands. These might be the easiest way, but you have to double these many times to get it small and tight enough that you need to tie off the ends so they're watertight. We also have little threads we could tie knots on the ends, or we got these which are, you know what that is? I call it a hair barrette, but it just clips on the end of that tube, and that they work pretty good. That's simple. You should use these, but any of those three ones. So we need to close off one end, and then we're going to fill that balloon with something. And, so what we're going to fill it with is sugar water. This is sugar, sucrose. So just mix some of this with some distilled water. That'll help me measure that, but that's too much. So we fill that balloon with the sugar water, and then the beaker, put it in the beaker here, just pure distilled water. And this dialysis tubing, water can pass through it, in or out of it, but the sugar molecules cannot. So we got sugar water in the balloon that's surrounded by pure water. So that means the sugar water would be hypertonic compared to the distilled water that would be hypotonic. Hypertonic means more stuff dissolved in it. Hypotonic means less stuff dissolved in it. So fill this with sugar water, tie off both ends, and then we weigh it. Forgot to get we got little electronic balances or scales back here. Need to get one now. So we'll weigh it, we'll pat it dry on the outside and then weigh it. And then we'll put it in here and let it sit for a week. What would happen if we?
Osmosis will get the high concentration. Right.
So osmosis moves the water from hypo to hyper, so it moves from the distilled water into the balloon, right? Because the sugar water is hypertonic compared to the distilled water. So if we weigh it, and it weighs, I don't know, 20 grams, and then a week from now we take it out, pat it dry on the outside, and weigh it. It should weigh more, right? So it should have absorbed water. The water should move into the balloon, right? If we would do it the other way, put sugar water in the beaker, pure water in the balloon, the balloon should shrivel and lose water. So I was saying we could do both ways. We can label on which one. Maybe sugar in the balloon, water in the balloon. And, so you need a couple of you, some of you guys to do it. So just we got 2 of these. So some of you guys work together. Somebody put the clip on it and hold it. Somebody needs to hold it open, while somebody else uses a stopper to push up to fill it with sugar water or pure water. Okay. So volunteers, who does it? I'm going to get the scales. So this one's going to be pure water, so it's going to have sugar water in the balloon. This one, we'll put sugar water in the beaker. So it'll get pure water in the blood. Go in there.
You have the, somebody who be.
Yeah, the Sharpies. Sharpies are in this drawer. If you have any Sharpies, they're in here. There's a bunch of them. So, droppers. I don't know if you want to use the clips, it might be easiest. So I'll get the electronic scales. I need at least one more helper. Does somebody else want to help? Use this to fill it. Yeah, you're they're gonna put sugar water in there.
Oh, okay.
A little more. You try, maybe another clip or something, and try to get all the air out of it. Put it right at the top of the water. So just pat it dry on the outside, but we only want to break what's inside. Maybe if you fold it over on that. We're going ahead and break off the clips.
It started off. It started off so pretty well. The other end.
So pat it dry on the outside and then you because we only want to wait what's on the inside, not the outside. And we'll put it up in the- Put these thin lines here too.
So this is sugar water and goes inside the distillery.
Yes. Yeah, that's sugar water, obviously. Yeah, usually sugar water stuff is in there. So we should write that down somewhere.
12.33, that's all 33.
12.33.
Yeah.
Regulations.
So like I'm kind of like it.
Yes.
Maybe we should just switch.
That water. Oh.
And that's not what it is. You can actually check it out.
Well, 17.35. The 20.5.
20.53.
It's the one with pure water in it. That's gonna go in the sugar water. When we come back next week, we'll get it out and weigh them, see what happens. Okay, well, you guys probably tired, you had an exam. Is it okay if we?
Yes.
Yes. I don't even have to say it. Yeah, that's fair. You have a good weekend. Come on. What is... Let's take 10 tries to do stuff.